recording. Get you fine. So today I want us to go through endemic and epidemic diseases and also see the difference between the two. So an epidemic is the occurrence of a disease case at a frequency population in a given period of time. For example, normally we do have some malaria cases now and then. For example, throughout the entire year, we do in our hospitals, we do have some cases of malaria being reported now and then, but there is reach a time where we have a lot of people going to the hospital or suffering from a particular disease, and especially the communicable diseases or infectious diseases. For example, cholera, we can have once in a while some cholera cases being reported, but there are times when we have a lot of cases being reported in the hospital or a lot of people suffering from that particular disease. So when we have this particular time where we have a lot of people suffering from a particular infectious disease, we refer to that disease as an epidemic because it is occurring with cases or frequencies that are higher than the normal being reported in a hospital or people suffering from that particular disease. Then we have another category of diseases occurrence, which is endemic. On the other hand, it is the occurrence of diseases or condition in a given population at frequencies that are constant over duration of time. So sometimes the endemic diseases can become epidemic. So when we have diseases that are people suffering from a particular disease at a normal rate, a not alarming rate, then we refer to that particular disease occurrence as endemic. When this endemic shoots, or when a lot of people are suffering from that particular infectious disease at a particular time, then we refer to that disease occurrence as an epidemic. So it is important to note that some communicable diseases are endemic in some regions of our country. For example, malaria is endemic in Coast. Malaria is endemic in Nyanza region because of the conditions that normally encourage the growth and multiplication of mosquitoes. But in some areas, for example, in central, in rural, for example, we don't have a lot of malaria being reported now and then because the conditions that are necessary for mosquitoes to thrive are not encouraging in rural. The same case in Thailand areas, we have very cold environments that are not encouraging to malaria growth or mosquito growth, sorry. So we have some areas where the endemic are, or these endemics or some diseases are normally endemic in some regions of our country, Kenya. So we can't rely, or we can't say that those diseases are epidemic because majority of people normally now and then report malaria cases in those regions. But there is a point where even in those areas where malaria is endemic, still that malaria can also be an epidemic in those places. For example, Baringo. There's reached a point where there's some season where we have a lot of people suffering and dying from malaria in Baringo. So at that point, we can say that the malaria there is epidemic because it is occurring above the normal frequency or the normal range of diseases being reported. So we have a diagram here. As you can see, the epidemic normally ranges or normally varies depending on the season, as you can see. But for the endemic, the pattern is equal or the pattern is normal throughout. But for the epidemic, we have variation in the pattern. There's some time where we have a lot of the diseases, there's sometimes where we don't have the diseases. So when you have a lot of these diseases, you refer to that occurrence as epidemic. When we have normal occurrence of the diseases, endemic. So we can say endemic or malaria is endemic in coastal region because people normally suffer from malaria throughout. But in some cases or in some areas such as Baringo, malaria can be epidemic because of the environmental variations. How do we investigate epidemics? So epidemics of infectious diseases, we have such as cholera, meningitis, typhoid, typhus, plague, are important threat in many countries. For example, cholera is a threat. Once you have a cholera case in an area, that calls a lot of attention from the epidemiologists, from the health workers. Each and every person is called to attention because cholera is a threatening disease in any region. So outbreaks of such diseases affect many people and are caused by infections, dietary deficiencies, or infections. So you'll find these diseases such as cholera being caused by infections, by Vibrio cholerae. When you have dietary malnutrition in a country, for example, kwashiorkor, marasmus is a very threatening conditions or diseases in any particular region. So modern epidemiology arose out of the studies of such diseases because they are a threat to any region. Then epidemics can be either categorized into point source epidemics or contagious epidemics. Let's see the difference between the two. So for the point source epidemics, in this type of epidemics, many susceptible individuals are exposed more or less simultaneously to a source of pathogenic organism or toxins. For example, if we can remember, there is reached a period where we have a lot of malaria being reported in Baringo County. So at that time, you find a lot of people being exposed to these malarial parasites at once or simultaneously at the same time. So you'll find a lot of people suffering from the malaria approximately at the same time. So then as 10 guests, as for example, we have exposure to a source of pathogenic organism. For example, let's take an example. You're invited in a ceremony, for example, a wedding ceremony, and the food that is prepared there have some toxins that may cause some infections in you. So you'll find a lot of people suffering from that particular, maybe food poisoning at the same time, because that was a point source epidemic. It came from one, or you were exposed at once. A lot of you were exposed at the same time. Another example is when guests at a feast eat meat contaminated with Staphylococcus aureus. So you'll find people suffering from, for example, typhoid because the meat maybe had some Salmonella typhi, a bacteria that normally causes typhoid. So this results in explosive increase in the number of cases of diseases over a very short period of time because majority of the people are exposed at the same time. Then another type of epidemic is contagious disease epidemic, and this is passed from one person to the other. So this type of an epidemic results when organisms are propagated in the community by passage from one person to the other, so that the initial rise in the number of cases is less abrupt than in point source epidemic. So you will find at the first or during the first period of infection, few people will have the disease. For example, the disease that we are trying to overcome right now, corona, we started with one individual in Kenya. Right now, we have a lot of people suffering from corona. So it started as a contagious disease epidemic, but now we are having point source epidemics because a lot of people are suffering from the disease. So the difference is that for the point, many people are exposed at the same time and they suffer from the disease at the same time. For the contagious, at first, these are the diseases that will be passed from one person to the other. So at first we'll have few people and then later we have a large number of people because the disease will have spread to a lot of people. The next part that I want us to cover is stages in the investigation of an epidemic. How do we investigate? For example, when we have a cholera outbreak in Ruiru, how do we investigate that? What are the steps that you normally follow in order to investigate the source of the cholera? Who is at risk of this cholera? How are they getting, or how is this cholera being transmitted from one person to the other? So how is this disease spreading? So these are the stages that you can follow. The first stage is verification of the diagnosis. The next stage is confirmation of the existence of an epidemic. The third stage is identification of affected persons and their characteristics in two ways. We have case histories, search for additional cases. Then the fourth one is definition and investigation of the population at risk. So I'm just mentioning them because the next thing is I'll explain each stage at a time. So the sixth one is formulation of hypothesis as to the source and the spread of epidemics. Then the second act is through management of the epidemics, through that is through treatment of the cases, prevention of the spread, writing a report, and continued surveillance of the population. Then lastly, you experiment, you verify the agents of the disease and the mode of spread. So let's go one step at a time. So the first step is verification of the diagnosis. And this is when information is received about a suspected outbreak. For example, you have maybe two people coming to the same health center with cholera. So you receive, when you receive this information of this suspected outbreak, it is important to take a detailed history from the informant, whoever is reporting this outbreak or reporting this cholera. You need to look for more detailed history about this disease. How many people have been reporting with this cholera in Ruiru sub-county, in Kiambu County? So a tentative diagnosis can then be made, e.g. cholera, food poisoning, etc., as this would enable the process of mobilization of resources, which includes manpower, materials and supplies, as well as finances and timing. So you need to do a lot of diagnosis of these people. Then this diagnosis need to be done in order for you to be able to be sure that there is an outbreak. And remember, an outbreak also requires a lot of resources in terms of people, in terms of materials and supplies, drugs, gloves, and whatever, as well as money, as well as the time to treat this outbreak. So laboratory tests and in some cases, out of this, of the people maybe who have died from the same disease, should be carried out as soon as possible to verify the actual cause of the epidemic. So you need to test more and more people. For example, if we have, right now we are dealing with corona. So majority of the hospitals, when you present yourself with suspecting symptoms, you must be tested in those hospitals. So at that time, they are diagnosing that type of disease that they're suspecting is an outbreak or has an outbreak, in order to ensure that, and also people who have died with the suspected symptoms are also tested in order to verify the actual cause of the epidemic. You can't know the cause if you don't diagnose first. You have to diagnose first. And by diagnosing, you are verifying whether the outbreak exists or not. For example, in Kenya, we wouldn't know that there is an outbreak of corona if at all they were not testing to verify that we have the coronavirus in Kenya. In Tanzania, they're not testing. So they will say that they don't have the corona because they're not verifying through diagnosis, through the testing. And the testing can be done to a normal human being or an alive human being or a dead body of a human being. So the second stage is confirmation of the existence of an epidemic. You need to confirm the next stage. So during the next stage, So due to the advances in laboratory tests and treatment, at times it may take a while before existence of an epidemic in a community is ascertained. So it is wise to check on the previous record with regard to any suspicious disease that may present at a health facility. So this may avert a crisis, as many infected individuals in the community can be identified or traced in time. Existence of an epidemic should be demonstrated by a graph of incidence against time and by mapping its geographical extent. So the beginning of the epidemic is assumed to be the earliest time the incidence rise above the normal endemic level. So during confirmation, you need also to include previously, for example, a 1 month prior to the time that you're suspecting there is an outbreak. You need to check those records of the people that have been reported in the hospital. Because if you don't look at those records, and maybe these people are presenting with cholera and they were misdiagnosed by the hospital, they will go and infect others. So you need to go backward and look also any suspicious diseases or any suspicious symptoms that were presented to the hospital at the previous maybe period, short period, maybe a month or 2 weeks or so, in order to make sure that whoever came during that period, they didn't go and spread the disease to population at risk in the community. So you still need to go and confirm back through your records in the hospitals in order to ensure that those who presented with symptoms that were suspicious and maybe they were not diagnosed with this particular disease that you're dealing with are also traced and treated if at all they have the disease. And this will enable to prevent the transmission further to the population at risk. Then the third thing to do is identification of the affected persons and their characteristics. You need to identify who is being affected and also identify also their characteristics. Characteristics can be in form of age, can be in form of where they are living, can be in form of where they are working, and many other characteristics that are personal. So identification of this individual and their characteristics is done in 2 ways. The first one is case histories. You need to look for case history, and details of each confirmed or suspected cases must be taken in order to get a complete picture of the epidemic. So the details should include at least the name, the age, the sex, the place of residence, recent movements, details of symptoms. So these are the details that we will normally go and give a person who is doing a triad on you in the hospital. You ask of how old are you, your sex, what kind of work you do, where do you stay, have you been moving previously, where have you been maybe moving from one county to the other previously, maybe 1 week prior. Then how are you feeling? What are you feeling? That is the symptoms and signs. And as well as dates of previous illnesses and immunizations. When did you start to feel maybe muscle pains? When did you start to sneeze? When did you start to cough? All of these are case history. So this information should be recorded on special cards, and if possible, especially if cases are more than 1,000, coded and entered in a computer for ease of analysis. So in majority nowadays of the hospitals we have in Kenya, we have computers or we have systems that normally capture this data. The next type of data that is normally collected during the identification of these affected individuals is search for additional cases. And these initial cases may be recognized in a hospital, hence there is need to visit the nearby dispensaries, clinic, homestead, so as to reach other cases that may be unidentified or treated. For example, if you're dealing with a very contagious disease, for example, corona, the health worker will still go and pick your neighbors around you, your family members who you are staying with in a a homestead, in order to go and look whether you left other cases that might be transmitting the disease to other people. And also, if you, for example, suspect cholera has been, people have been presenting with corona cases in Ruido Sub-County Hospital, you still need to go to other nearby hospitals in order to look for other people that have been reported into those hospitals with maybe the cholera, and then that's how you can be able to trace them and be able to treat them to avoid the transmission to other population at risk individuals. Then the next step is definition and investigation. That is the third step, definition and investigation of the population at risk. Now that you have been able to verify the diagnosis, you're able to confirm that there is disease in a particular region, and you're able to identify the characteristics of the individuals reporting with this disease, then you need to go and look for, using the details or the details that you have captured from the previous three stages, you need now to go and look at the characteristics that have been presenting in order for you to know who is at more risk of contracting this disease, who is at more risk of getting corona, who is at more risk of getting cholera, who is at more risk of getting malaria. And then you can be able to come up with the mitigation interventions. So definition and investigation of a population at risk includes analysis of case history. You need to analyze the characteristics of the individual who have presented with this disease, and this will give a profile in terms of age, in terms of sex, in terms of residence, in terms of occupation. You might find that majority of people presenting with corona are the people maybe working in airports, working in restaurants, working in bars, working in a particular area. The people, or more people are presenting coming from slums, more people are presenting coming from a particular region, more people are presenting from Isli, more people are presenting from Mombasa. So this characteristic will enable you to know who is at more risk of getting this disease and how can we help these individuals. So at this level, an attempt should be made to calculate the disease attack rate, that is the number of cases in the population at risk at a specified duration of time. That is attack rate. Remember types of incident rates. This is an example here. So it is important to compare the characteristics of or behavior of a case that is affected with that of cases that are unaffected. So this will give you a clear picture of who is at more risk of getting the particular disease. So this would bring out a unique profile among the affected, e.g., type of food eaten, the source of water they normally consume, the place of work they normally live. So investigation of the population at risk should be conducted in order to identify those in the asymptomatic stages, carriers, and non-affected individuals in a population. So by investigating those that are at risk, you'll be able to know, since we are saying that maybe this kind of, or this age, or this sex is more at risk of getting this particular disease, then we can also go deeper and investigate the characteristics of that age group, the characteristics of that age group, or maybe gender group, or even the type of the diseases maybe they had. If you're dealing with people suffering from hypertension and they're at more risk of getting a particular disease such as corona, you need to go and investigate what is this particular characteristics do these people at risk are having that are putting them more at risk of getting this particular type of disease. So by that, you'll be able to know who is asymptomatic, who is not having any symptom, but they're carrying this infectious pathogenic in them, pathogenic microorganism in them. Who are carriers? They cannot be affected by the disease, but they carry the agent of infection. Then, who is more affected and who is not affected by this kind of disease in that particular population at risk group. So the outcome may determine those who may need isolation or segregation for ease of disease control. So this information is the one that is guiding people in preventing the spread of this coronavirus. Some people are being isolated because they are asymptomatic case, but they are asymptomatic case, but they still harbor the infection in them. So the outcome may determine those who may need isolation or segregation for ease of the disease control. So this stage involves the identification of those who are at risk and also identifying the characteristics of the people who are at risk. And this will give more know-how on how to manage this infection. Do we need to segregate them? Do we need to isolate them in order to overcome the spread of this infection? So the next stage is formulation of hypotheses as to source and spread of epidemics. So you may need to know where the epidemic occurred, when it did, and the prevailing condition for it occurring. So to formulate a hypothesis normally helps to research further or create like a question in order for you to research further about the particular disease. And this will guide people to know when did corona occurred, when this cholera outbreak occurred, and when did it start, and what are the conditions that were necessary or that were prevailing or that were existing for this corona to occur. So ideally, before an epidemic occurs, it is important for those concerned, especially the health workers, to assess the crucial influences that, when altered, would ensure the epidemic does not rise. For example, seasonal cases of cholera or malaria can be averted if the trends are known and can be altered prior to the epidemic's occurrence. But what happens in Kenya? You'll find each and every year we have seasons of malaria in Baringo, seasons of malnutrition in a particular area. But it's upon those who are responsible, that is the county health workers, as well as the governor himself, together with his ministers, to make sure that mitigation measures are put in place in places where an epidemic is suspected to occur. For example, they can spread mosquito nets, they can pellet people to clear bushes around them. But what happens? They just let it to happen in order for them to be able to walk around with some money when they're trying to help this individual. But what happens? People die of malaria, and this is something that can be prevented. This can be something can be averted if the trend is well catered for, or if the trend is stopped from happening. When formulating a working hypothesis about an epidemic, the following must be considered. When you want to come up with a hypothesis, we have a research question and we have a hypothesis. We can develop a hypothesis. For example, females are more affected by corona than male. That is a hypothesis. You can also say, you can also phrase it in a null way, where you're saying females are not affected as compared to men. Or we have more females suffering from corona as compared to men. The negative of it is we have less females suffering from corona as compared to men. So that's a hypothesis that you need to create in order for you to be able to go deeper and look, is your hypothesis correct or not correct? Are you able to reject or not reject the hypothesis? So the following are normally considered when you want to formulate a hypothesis. The first one is the agent of the disease and its characteristics. You need to identify what is causing, or what is the cause of this, or what is transmitting, what is being used as the vehicle of transmission from one person to the other of this disease. Is it a bacteria? What kind of bacteria are you dealing with? Is it a virus. What kind of a virus are you dealing with? Is it a fungus? What type of fungi are you dealing with? The second one is the receiver. That is the human being as a case of carrier, other animals who is carrying this or who is spreading this. Is this a disease being spread through water, through soil, through animals, through human beings, through birds, through objects? Corona is passed through human beings. It can also be passed through chairs. It can also be passed through objects. It can be passed through iron. It can be passed through wood. The next one is the mode of exit from the receiver or source. How is corona moving from one individual to the other? Is it through tears? Is it through droplets? Is it through fecal matter? Then the mode of transmission to the next host. Is it transmission through food? Is it transmission through water? What is the mode of transmission? Is it through droplets? Is it through fecal matter? The mode of entry, for example, corona. How do corona get into a human being? Through the mouth, through the nose, through the eyes. Then the susceptibility of the host. How susceptible, how quick do the hosts able to transmit this infection to another person? So this will be included when you're formulating the hypothesis. And this hypothesis, remember, the one that will help you have some answers to the spread as well as the cause of this type of infection. Then the next step is management of the epidemic. Now that you have diagnosed, you have confirmed, you have done all these stages, then how do you manage this epidemic? Number one, you treat the cases that are being presented now and then. By treating, you're helping this individual suffering from the same disease, as well as you're also helping this individual, or you're preventing the transmission of this disease from the individual suffering from the population at risk. So following identification of cases, healthcare workers have the responsibility to treat them. So some condition like cholera may require arrangement first. You need some specific type of beds because they are not able to move from that bed to loo or to the toilet. So you need a specific bed that will enable this person to pass through easily. You need some accommodation. You can't take them to the normal ward because they will continue to transmit the disease. You need a specific training, or you need training to the staff. Whoever is cleaning that area, the nurse, the doctor, still needs some special training. If you're dealing with students, you also need to train them. So if you're dealing with an army, you need to train them. You need to train, for example, corona. When corona reached Kenya, all medical staff, including the person at the gate or the guard who is welcoming people in the hospital, were trained on how to prevent themselves from getting the corona from whoever is coming to the hospital. All the health workers were trained on how to overcome the spread of this, or even to treat this type of corona. So that's the first thing. So estimation of required supplies may be done for purpose of procurement or acquisition through borrowing from nearby health facility. Rehabilitation measures may be needed for certain diseases such as polio. You still need some rehabilitation. For example, polio, when we have a polio outbreak, a lot of people will be disabled in one way or another. So you still need to rehabilitate them even after you treat them. So the second thing under management of the epidemic is prevention of the spread and commencement of control measures. So once the disease epidemic has been identified, it is possible to plan an effective control program. So, however, even before the epidemiology is worked out, the healthcare workers should try and institute simple measures to minimize the spread of the disease. So we need to come up with mitigation measures. We need to come up with intervention measures in order to prevent the spread of these infectious diseases. Otherwise, we'll continue to live with the infectious diseases forever. If it is prevention through immunization, if it is in prevention through isolation of the cases that are suffering, if it is through health education, if it is protective barriers or protective gears, use of these protective gears, you need to incorporate them in order to prevent the spread of this infection. So community must also be mobilized and given more health education in order also for them to prevent themselves from getting the disease, and by that you are preventing the spread of this infection. You need to, people are nowadays being educated through the TVs, through the radios, through our churches, through a lot of groups, how to prevent themselves from getting corona. And by empowering them to overcome the spread of the corona, you're preventing the spread of this corona among the community members. Then still the third, still under management of this epidemic, the third thing to do here is report writing. And these are usually three types of report. There are usually three types of report to be written in an epidemic management situation. The first one is popular account. And this is for lay people so that they understand the nature of the epidemic and what is required of them to prevent the spread or reoccurrence of this particular disease. So you need a report in order to educate the people that are affected in by a particular disease, to educate them more, maybe later in future. That's why we have to write a report in order for people in a particular region suffering from a particular disease to overcome right now, also to overcome later in life. Another thing is planner's account, and this is for especially the government and the local authorities, so as to ensure that all the necessary administrative steps are taken to limit the outbreak or to prevent reoccurrence. You still need to give this report to the administration or the local authorities that are responsible for these individuals. Once the chief has this report, he'll be able to enact more measures in order to prevent the cholera from happening later in life. For example, maybe a cholera outbreak came from a certain hotel. So this chief will ensure that the hotels in that region maintain maximum cleanliness in order to overcome the occurrence of that corona again in life. Then you have scientific report. This is for publication in medical journals, for sharing with other scientists, and also teaching purposes. It will also give more insight into more research into that particular disease in order to get more solutions to overcome the reoccurrence of the disease in future. Still under management of the disease, we have continued surveillance of the population. You don't leave the population alone. You still need to do some follow-up in order to ensure that whatever you taught them during the outbreak, they are still practicing even after the outbreak is over in order to overcome the occurrence of the particular infection. So when do we get sources of surveillance information? Number one is notification of the illnesses by medical personnel, auxiliaries, chief, employer, school teachers, head, families. This will give you, do we still have some cases even after the outbreak is over? Do we still have some cases being reported? Then, and this will help you know, or whoever that will give this information is through the hospital, through chiefs, through employers, through school teachers. They will be able to have this information. In our school, do we have this cholera? Still, do we have still children suffering from this cholera, even after the outbreak is over? The second one is certification of deaths. Some people will still die of the cholera later in life. So you can also get information from the deaths. Then data from sources such as public health laboratories, as well as entomological and veterinary services. So you still get this information from these people. So all these sources are important and relevant for effective surveillance. You need to follow up in order to ensure that people are practicing prevention measures, and also to know whether there are still people being reporting with this particular disease. And this information will get them from the first source, the second source, as well as the third source. Then the last step of epidemics, investigation of these epidemics, is experimental verification of agent of disease and mode of spread. So experimental evidence may be needed to verify the hypothesis about the epidemic disease causative agent. So you need to experiment in order to be sure that this particular infection or this particular epidemic disease is caused by a particular agent. So you're now trying to verify the hypothesis that you set in the previous step. So one may want to prove that meat can be contaminated by a contaminated meat slicer. You are verifying the hypothesis. Maybe you came up with a hypothesis that this infection is being brought about by, is coming from a certain butchery. And this butchery, or the main source of this infection, is this particular spade that is being used to cut the meat. So that is the hypothesis. So you still have to come back and experiment. Do this particular spade have this particular agent that is causing this disease? So you need to experiment in order to ensure that that particular slicer that maybe is not cleaned now and then is the source of this contamination. That particular water that is coming from a particular stream or particular river is the source of a particular bacteria that is causing a lot of infection in a particular village. So you are trying to verify the hypothesis that you set. Maybe you set, or maybe you came up with the hypothesis that cholera is being caused by the water that people are consuming in a particular village. That is a hypothesis. So you still need to come back and verify, is this or experiment, do this water have this particular Vibrio cholerae? So such experimental studies may take long to come up with the finding, and hence may be completed long after the epidemic has been controlled. But it is important because it will help you to control the epidemic if in case it happens in another area or in that particular area in future. So that marks the end of this class. So go through these steps. Remember, we have two types of diseases. You have epidemic and endemic. Epidemic can be either a point source or a contagious. And remember these steps that are normally followed in the investigation of an epidemic. You verify the diagnosis. You confirm the existence of an epidemic. You identify the affected persons and their characteristics through case histories, such of additional cases. Then you define and investigate the population at risk. And this will give you more insight in the formulation of hypotheses as to the source and the spread of the epidemic. So this hypothesis will help you know the cause of the, or the source and the cause of the particular disease and how this disease have been spreading. Then you manage the epidemic through the treatment, through prevention, through writing reports, and also continued surveillance of the population. Then lastly, you have to test the hypothesis that you set here in the fifth step. And testing this hypothesis will give you more clear picture on both the cause as well as the spread of this particular disease. So those are the steps that are normally followed during investigation of an epidemic. So I hope these have really helped you, and I hope you will follow it up. So thank you and all the best.